All right, let me continue for the second part of this lecture on Chapter 16, Section 2, which is about prokaryotic organisms. We're focusing on bacteria. And I talked a little bit last time about the structure of a bacterial cell. And we were just finishing up on the shapes of bacterial cells, and I mentioned that there are three primary categories of shapes. There's some bacteria that don't fall into this category, but nonetheless this is a major way of classifying bacteria. Cocci are bacteria that are round. They can exist as individual cells. Sometimes they form couplets, or sometimes form chains, or even clusters. The spiral bacteria are called spirochetes, and they tend to look something like that. And lastly, the bacteria that are rod-shaped are called bacilli. And again, they can also form individual cells, or you might sometimes see them as connected end-to-end to form a chain. Another characteristic of the bacteria cell that's really important in diagnosing microbial diseases and categorizing bacteria is the cell wall. So we're going to take a look at the cell wall structure and a type of staining technique that's used to distinguish different categories of pathogenic bacteria. Okay, as I mentioned before, almost all bacteria have cell walls. There's maybe just a few that do not. The function of the cell wall is to help maintain the shape of the bacterial cell. Also protects the cell, particularly in environments like a hypotonic environment, in which water would tend to try to diffuse into the cell, could potentially lyse the cell, so the cell wall is there to protect it. There are two categories that are recognized regarding the structure of cell wall. And we sometimes refer to them by how they react to a particular staining technique that's used by microbiologists. And this staining technique is called a Gram stain. It was first used by an individual by the name of last name of Gram. And the reason that this type of stain is important because it actually helps physicians determine what kind of antibiotics to use to treat a disease. So it's kind of a valuable tool in medicine. A Gram stain consists of two stains that are used sequentially. And one is called crystal violet. So when the bacterial cell reacts with it, if it does, it produces a dark blue or violet color. The second stain is usually a pink colored stain. Safranin is sometimes used, and this produces cells that stain a pink color. All right, so let's take a look at the two types of cell walls. And I'm going to distinguish these by the reaction to the gram stain first. So we have cells that are called gram positive, and they are those that react with the crystal violet. So they are violet, very dark colored. These cells tend to have, of course, a cell membrane. So cell membrane. And surrounding that is a cell wall that is composed of a substance called peptidoglycan. And this is a molecule that contains some carbohydrate as well as some amino acid protein type residues. There's a second category of bacteria that we call gram-negative bacteria. They actually stain kind of a pink color. And their cell walls are a little bit different. They have, of course, an inner cell membrane. They have an outer layer of peptidoglycan, and on top of that they actually have another layer of lipids that we call the outer membrane. So these cell walls are a little bit thicker than the gram positive, and as I mentioned before, the structure of the cell wall, the differences are important because if we have a gram negative infection, we have to use certain types of antibiotics. If we have a gram positive infection, we have to use a different category of antibiotics. How am I doing on time? Oh, okay, I'm sorry. Okay, let me cut it there.